All right, welcome everyone. I think we're still letting some people in. Okay, welcome everyone. Better. Welcome everyone. Good morning, good afternoon, good evening, wherever you're from. Welcome to today's webinar. It is a joint webinar between Triskelion and Linge Agroconsultancy. The topic for today is from peptide discovery to product approval. Without further ado, yeah. All right, perfect. Thank you. I think I was on mute. Okay. But yeah, welcome everyone. Sorry about the technical issues before. Welcome to today's webinar about from peptide discovery to product approval. This is a joint webinar between Triskelion and Linge Agroconsultancy. So we are part of Certania, so we are sister companies. And without further ado, let's discuss about the webinar technicalities. So this is going to be a 30 minutes webinar. And there's a Q&A button on top of the screen, the shared screen. If you have any questions that you would like to ask, you can ask it via the Q&A button up there. And unfortunately, the questions that we would like to cover during the webinar is only the pre-selected questions. So any questions asked during the webinar will be answered via email, post, or after the webinar. So this is our first speaker for today, Dr. Rogier Kolnaar. He has a PhD in agricultural sciences and MS and master's degree in crop protection. He has 20 plus years in regulatory affairs and has been consulting since 2006. He is the registration manager at Linge Agroconsultancy. Second speaker for today is from Triskelion, Dr. Kleinnijenhuis. He's a senior scientist in bioanalytical chemistry and has been working or has industry experience since 2007 from TNO up until today's TNO spinoff, which is Triskelion. And he is also the specialist in peptide LCMS. All right, so this is the first presentation from Rogier. The stage is yours.
Next slide, please. Good afternoon, everybody. Well, I'll discuss at least a part of the whole regulatory affairs, what you could be dealing with when you're dealing with peptides and proteins to increase sustainability of plant protection. There has been a lot of research which resulted in quite a number of product developments based on peptides and proteins, such as plant elicitors, herbicides, fungicides, insecticides, that kind of products, and most probably more. These products can be based on antibodies, enzymes, neuropeptides, antimicrobial peptides, and most probably a lot of other peptides and proteins that you can imagine. One of the reasons research resulted in the use of peptides and proteins is that they are naturally occurring, and they can be quite effective, of course. This can be quite important for your product registration to keep in mind. There are different ways to describe peptides and proteins, and the division in general is made between natural ones and natural-like, that are the naturally occurring substances, or substances that are a little bit changed by human, but not to a large extent. They can still be considered natural substances, which I will discuss later, is also considered biopesticides. You also have the so-called unnatural proteins. These ones are completely developed by human, synthesized, and they can also exist of amino acids that are not naturally occurring. Next slide, please. Looking at the regulation, generally speaking, peptides and proteins are considered chemicals. Having said that, of course, they are not the normal chemicals that the plant protection products industry is working with, but these are as they are strings of amino acids connected with peptide bonds. It's just different than all the other stuff. For the legislation, there will be an update at the end of this year, and there most probably peptides and proteins will be included in the definition of biocontrol active substances, which has certain advantages. If the proposal is accepted, this might mean that your product could get provisional authorizations already during evaluation of your substance, and EU promises a fast-track procedure. Of course, we have to wait and see how that will happen, but at least there is some change ongoing. Next slide, please. As already explained, the substances are considered chemical substances by Europe. So if you go for the data requirements and you check them in the relevant regulations, you will stick to Part A, and you will already quite soon find out that a lot of data requirements may not fit to your substances. Unfortunately, there is no guidance document available, at least not an approved one by EU, but the IBMA and the CropLife EU have both developed separately from each other decision trees, which may be very helpful if you address your products. The one from IBMA is published by Marloes Buscher et al. In 2023, and quite recently the one from CropLife Europe was published by Criollo. IBMA one is mainly based on natural substances relating to plant extracts, etc., but they fit perfectly for peptides and proteins. The chemicals is a bit more broadly, but also fits perfectly. Then the advantage of peptides and proteins is that they are definitely not new in other kinds of industry and other legislations. So if you start with your dossier, make use of the legislations for GMOs, pharmaceuticals, cosmetics, feed and food. And you'll find a lot of information there. You'll find a lot of guidance, mainly on toxicology, unfortunately, but GMOs also have to address safety of environment. So there you'll find also some interesting stuff. Then finally, of course, there is a huge amount of scientific literature available on peptides and proteins, also addressing the legislations, mainly on pharmaceuticals and GMOs, which is very helpful and also provides specific information on what to do on your testing. Next slide, please. Well, unfortunately, there is no standard approach, at least not yet, and this is basically true by the fact that we are talking about a wide range of different kind of substances. I mean, the only thing peptides, proteins have in common is that they are based on amino acids with peptide bonds, but that's almost it. So first step of your registration of your dossier is just find out what you are dealing with. What is your active substance? What does it do? How do you produce it? Is it fermented by a microorganism or in another way? Then keep that organism into account because it might be necessary for your dossier. Do you synthesize it by in another way that may make it easier for sticking to the chemical requirements? Will not by definition make it easier for you. Purity, of course, is also quite important. If normal, well, at synthetic chemicals, purity should be as high as possible, but for a peptide and protein, you're able to specify it as a natural substance, which makes a lot of ranges possible for your purities. Analyze it well. You have to get a stable production method, so you have to provide a good 5 batch analysis, and Anna will discuss on that a lot more after I'm finished talking here. Other thing to keep into your mind is the mode of action. Are you working with a venom, for example, then you automatically know that the next point will be very important. If you're dealing with a non-toxic mode of action, you can work another way around. So for toxicology, work with a weight of evidence. Is it a food substance you are dealing with? This might most probably save you a lot of testing and a lot of literature to be searched. If not, then you'll have to start from bottom. Find out as much as information about your substance. Check if toxicology is mode of action or might be something that you haven't thought about it. If you have analyzed your literature review and you do not have sufficient information, check the guidance documents of, for example, pharmaceuticals, and you have a huge battery of in silico testing, in vitro testing, and in the worst case, if you really cannot avoid it, you can continue with in vivo testing. Before doing so, always contact the RMS because maybe they come up with some other thoughts, or at least you have approval to do so. Of course, for the risk assessment, human exposure stability is very important. Here you can take into account natural occurrence, and on that way you can prepare your waivers. Almost forgetting, allergenicity, unfortunately, is quite an important issue with peptides and proteins. Even if you have a food substance, I think most of you are aware of the fact that you can use milk freely as an antiviral agent, but please only clean your knives with it and do not start spraying, because that's asking for big problems. Next slide, please. Then the next important point, I skip residues here, but it will be addressed later during the Q&A, because to my opinion, residues is not the most important part of your dossier for peptides and proteins. E-fate might be an important one. Generally speaking, both peptides, proteins are expected to degrade quite rapidly into the environment. In general, they are food substances for other organisms. Organisms are in generally also used to feed on these substances. But you never know. It might be natural occurring, but when it's toxic or when it's a bit more complicated, or when you have a peptide or protein that is really really unstable, you might have formulated to stabilize your product, and then of course it will stay longer in the environment. So don't assume that it will degrade rapidly. Don't assume that as it is a peptide or a protein, it will act like all the others. Come with a proper justification. So check literature, check the molecules itself. Also, of course, check to which extent it might enter the environment, because when it's not that much, that might of course help. In the end, the residues in your that will be the result of degradation won't be a big issue. There might be some steps in between, but in the end, you're dealing with the individual amino acids and maybe some other molecules available. It all needs to be checked, so don't take it for granted, but most probably that's not the most important issue to be addressed. If you still, if by checking all the information you cannot avoid potential exposure to non-target organisms, yeah, then basically the same story is valid for ecotoxicology as is for toxicology. That means first check your substance, check the mode of action, dive into the literature, and if that's not enough, it's not in the slide, but here also you can start with the in silico testing first. There are models that you can use for various non-target organisms, not for all, always test it. If that's not sufficient, there may be some in vitro tests, not that much, so most probably you'll end up with in vivo testing then. And also take into account, even if there is no exposure, you still have a bottle or a package with your peptide or protein in it, so that might need some classification. So from that point of view, even if you think there is hardly an exposure, you should not neglect the ecotoxicology part. Next slide, please. Just to wrap up the story, unfortunately, no standard approach for these substances, so you have to develop your own strategy based on the knowledge of your own peptide. So not the general information, but really search for information for your own one. If you have your strategy and if you have some basic information, call the RMS and go for a pre-submission meeting and try to convince them from your strategy. They do not have a lot of experience with these kind of substances. In general, they are open for your proposals, but you have to explain them a lot. You have to talk with them a lot. And in the end, they won't be that bad, and they will hopefully start the evaluation soon. And for all this information, for all these strategies, of course, both Triskelion and Linge are there to help you. And I give the floor to you again.
Perfect. Thank you so much, Rogier. That was a beautiful presentation. So now we're on to the second presentation, which is about molecular barcode and targeted LCMS analysis, which will be presented by Anne Kleinnijenhuis. The floor is yours, Anne.
So good afternoon. Thanks for joining. Today I'm going to discuss data analytical workflow for complex samples, starting with non-targeted analysis, and then at the end focus more on targeted analysis of peptides and proteins. So there are many products which have complex molecular composition. Usually they have a biological origin, but they can also be heterogeneous chemicals. So if you produce batches of your product, how are you going to compare their consistency and quality? Of course, there are very general techniques like visual and olfactory, or you can maybe use infrared spectroscopy if you're more interested in the several plant or animal species which are present in your product. you might look at the DNA using next generation sequencing. Well, there's another option, non-targeted LC HRMS. It's a liquid chromatography high resolution mass spectrometry. It provides very detailed information at the molecular level of all the components of your mixture, but at the same time it can be used to more globally investigate similarity between samples. Next slide. So to explain more about analytical approach, of course we start with a sample or a process sample. Then we submit it to non-targeted analysis, and then we can perform non-targeted data analysis and slightly focus more towards targeted approach. Over here I also put a reference to an article we wrote about this. Next slide. So first, if you're dealing, for instance, with a plant material or plant extract, you first need to dissolve your sample or maybe apply a suitable extraction. Then we perform the analytical run and integrate the relevant part of the chromatogram to an MS spectrum. So we then already reduce the size of the data set a little bit, and then we export the MS data as intensity per nominal MOVC value and calculate the correlation coefficients between all the different samples. This approach you can, for instance, use to compare batches of your product. And then we transpose the intensities in a horizontal direction. Next slide. So over here I put some example what you get when you compare quite complex products. Maybe focus a little bit on the top left now. There are some food products which all contain soy as a component. And well, we get very high correlation coefficients already between these products. So they're actually quite similar. But yeah, of course, the appearance when they are in the supermarket or shop, the appearance is quite different. Next slide. So then when we go to the barcode, you already see in one view also similarity between complex samples and also differences. So you can use this approach to get first a global overview over your complex samples, and then in the barcode illustration already try to investigate where important differences or similarities are between samples. Next slide. Just as a second example, we received a protein hydrolysate sample which had an unknown processing, enzymatic and or chemical processing. And we compared it to several references, A to E. In the correlation coefficient matrix, you can already see that the sample X has highest similarity to reference E because it has the highest correlation to the sample. And also in the molecular barcode view, you can easily see the similarity between this sample and the reference E, just to illustrate what you can do with it. Next slide. Over here we analyzed a plant protection product, which was a plant extract as well. Because there can be many compounds in this sample, we analyze it in a mass spectrometer negative mode and in positive mode. So you actually get two comparisons between all the batches. In this case, the 5-batch analysis showed that the products were quite similar, but they contain many, I think, thousands of compounds. It actually is similar in the negative mode, which focuses more on detection of acidic compounds, and in positive mode, which focuses on more basic compounds. Next slide. So when you see in this global barcode view differences or similarities between samples, what do you do next? well, we still have the underlying data set, which contains high resolution mass spectrometry data and also fragmentation data of all the components in the mixture. So we zoom in a little bit on the barcode, and then we focus now on this green band. When we then look deeper into the data, let's say a dominant signal was detected in this channel. The products that were compared all contained bovine dairy as a component, yogurt gum, milk powder, and a dairy-based burger. And well, this compound, which was present in all the samples, actually originated from bovine alpha S1 casein. So it's quite a logical component, actually. So next slide. Wait a minute. So go back. So you can use it to compare samples, and also if you focus more on peptides in these data, the peptide sequences can be found, and also modifications of peptides can be mapped using a database search software. Next slide. I just wanted to show you we have quite a dedicated application called semi-targeted analysis. And we can detect, and it's like an analytical module within this overall approach. Then we can detect animal material in samples, let's say generally, because it focuses on a very specific collagen fragmentation, which occurs in the samples. Next slide. Okay, so when you already know a bit more about the proteins and peptides in your sample, of course, the next question is how much of these components is present in your sample. So then, of course, it's important to go towards targeted analysis, to quantify these components. So typically at Triskelion, we of course, when the matrix is very complex, it can help to do a selective purification to pull out your protein. One of the approaches to do that is to immobilize antibodies on magnetic beads, and then you can pull down the protein of interest. When we then go to the LCMS part again, we just cut up the protein and select generic or unique peptides to quantify an individual protein or maybe a protein class. Then we also synthesize a stable isotope labeled equivalent of such a peptide target. And then we can use it to really correct for matrix effect and response shift in LCMS to really obtain a quantitative method. Of course, this is not the only option to purify samples. This is one of the most specific ones, I would say. Next slide. So I presented to you about molecular barcode, and then later going towards targeted methodologies. We have used this approach for several subjects already. So to compare product batches of protein hydrolysates from milk and collagen and plant extracts. Also, we compared whole food products, and we use it also to assess changes in complex mixtures. For instance, during a production process or when a sample passes through an in vitro model. That's it. Thanks a lot.
All right. Thank you, Anna, for the very insightful presentation. I see that we're nearing the end of the webinar. I hope you still have time to stay tuned for the Q&A time. Unfortunately, due to time, we're only going to cover some of the questions that you have already posted during the registration of the webinar. So we will start with the first question here, which is how to specifically quantify large proteins, especially where reference standards are not available.
Yeah, this is a very important question when you want to quantify proteins. Of course, in the most ideal situation, you would have a certified reference of your protein. You can use this in your calibration samples. And then we also have the stable isotope version of our target peptide, which comes from the protein sequence. This is the most ideal. But often, there's not really a protein reference available. So next option would then be maybe to purchase an analog of your analyzed protein. It can also help during quantification. And if this does not work, then we fully go to peptide standards. So we cleave the protein using enzymes, and then we purchase peptide standards with a full purity statement. Of course, then we also need to assess the digestion efficiency, and we can do that. Just searching for peptides with miscleavages. So there are many options available depending on the materials that are available.
Okay, thank you so much, Anna. Let's move on to the second question here. How the evaluation is done by the authorities and studies to be provided.
Thank you. Well, actually, there's not a lot of experience there, not by the authorities, not by companies. So this is mainly based on the standard requirements for synthetic chemicals, and then it's up to you to convince the authorities that they should look at it in a different way, and that certain requirements are not relevant for your substances, and come up with good explanations why you think so. So that's basically the only answer I can give at this moment.
All right, thank you, Rogier. We still have a couple of questions. Maybe we can, maybe this is going to be the last question here. But for peptide MRLs and or residues, can we rely on published literature for some requirements or request a waiver?
This depends on depends again on the properties of your peptides and proteins. Most of them, or no, do not say that correctly. A part of the group is just digested easily in the body, which will result in amino acids or maybe some larger products. So from that point of view, I would not expect any residues that are relevant for MRL setting. And in addition, of course, after application of your product, it will most probably degrade quite fast. So yes, request a waiver, but a request one based on information that you have available on your own substances, and of course, published scientific literature is very useful here. I hope that answers the question.
Thank you, Rogier. And last but not least, I think it's also important to go back to the question 3, which is the analytical standard for quantification, because this came up in the question section during the registration form. Maybe Anna, you can help answer this. Anna, I think you are still on mute.
Yeah, I already answered it during the first question partly. So ideally, you have a certified analytical standard of the protein, but if it's not available, you search for analogs of the protein to serve as standard. And yeah, in many cases, these are also not available. So then it's good to perform the bottom-up approach, let's say, so digest your protein and then synthesize peptide standards. So at the end, we will come there, but it depends on the materials which are available for your protein.
Okay, thank you. Thank you, Anna. And once again, thank you for both of you, Anna and Rogier, for the presentation and the answers to the Q&A submitted during the registration form. And last but not least, before ending the webinar, if you have any more questions, feel free to add it into the Q&A button on top of the shared screen. And if you still have more questions or information that you would like to know, you can please get in touch with my colleague here, Isabel. Her contact detail is listed down below, which is isabel.vanlanen@triskelion.nl, and the phone number is provided here as well. So once again, thank you so much, everyone, for your time and joining the webinar for today. And we hope to see you again in our next webinar. Thank you so much and have a good one. Bye.